Well, welcome back, everybody, from the break. In the earlier session, we covered some important sort of concepts in terms of the populations that we get into our trials. And we also covered a perspective on endpoints, which is how we judge the outcome of an intervention. Now, for this session, our first talk will be on statistical principles for clinical drug development. This is really how we analyze trials, and I'm very delighted to welcome Mark Levenson, my colleague here, to give that talk. And that will be followed by a talk on another important aspect of trials that we haven't touched on yet, which is safety considerations in clinical drug development. And Shabnam Naseer will be giving that talk. I think that will sort of give a more comprehensive view of how we look at efficacy and safety. So without any further ado, let's turn over to Mark for discussion on the statistical principles of clinical drug development. Questions will follow the two talks. Thanks.
Hey, so good afternoon, everyone. My name is Mark Levenson. Today I'll be presenting on statistical principles for clinical development. This talk will be very foundational on principles that I hope will serve you well as investigators or even when you're reviewing other clinical studies. Here are the learning objectives. The first objective is rather broad: to understand basic statistical principles relevant to clinical studies. More specific objectives are to understand the concepts of bias and variability, to correctly understand p-values and hypothesis testing, to understand issues around multiplicity. The first point I wish to make is that design and conduct are more important than analysis. I will define these terms in a moment, but in other words, analysis cannot make up for poor design and conduct. Focus on good design and conduct, and the analysis will be straightforward. For the purposes of my presentation, I'm going to divide a study into 4 stages. The first stage is the design. This is the conception, planning, and specification of a study. This likely results in a protocol, a statistical analysis plan, investigator documents, training documents. The next stage is the conduct. This is the actual running of the study. Includes enrolling patients, treating patients, following up patients, obtaining endpoints, as well as monitoring study and data quality. The next stage is the analysis stage. This is the stage where the data collected in the conduct is converted and number crunched into actual findings that address the design of the study. And finally, there's reporting. This is a very important part of a study, but I won't have much to say about it today. Note that many people might think statistics only comes in the analysis stage. However, as I alluded to on the last slide, statistics is even more important in the design and even conduct stages. Okay, in order to market a drug in the US, you have to demonstrate a number of things. One of the most important things, of course, is to demonstrate efficacy. To demonstrate efficacy by law, you need substantial evidence, and that substantial evidence comes from adequate and well-controlled studies. There are regulations that spell out what an adequate and well-controlled study is. There are 7 elements which I briefly state here. Beyond their legal role, there are important scientific principles for all studies, whether you're going to submit a study to FDA or not. The first element is clear, objective summary of methods and results. So naturally, the objectives of the study should be clear. The methods you should use are going to be clear, and finally, the results should be clearly presented. So this is done in both the protocol and eventually the clinical study report. The next element is that the design permits a valid comparison with the control. This is to isolate the effect of the drug from other effects, like the natural history of a disease. For example, people may get better or worse on their own. The regulations talk about several types of controls, including concurrent controls, but also talks about historical controls, which may be relevant in certain clinical settings and probably becoming more relevant or more common nowadays with the interest in real-world evidence. The next element is adequate selection of patients. This may seem obvious that, the patients have the condition that you're trying to study, but it's sometimes not obvious. And again, in real-world evidence, sometimes this could be a little challenge to ensure that the patients actually have the condition you're trying to study. Assigning patients to treatment and control groups to minimize bias. This is typically done by randomization, and I'll talk about that in a moment. But the regulations say it doesn't have to be done by randomization in certain settings. For example, again, with a historical control group, there's no randomization involved. The next element is adequate measures to minimize biases on subjects, observers, and analysts. This has to do with blinding. We all know that if people know what drug they're on, the outcome might be different. If the investigator or the healthcare provider knows what drug a patient's on, that may affect the clinical management, and it may even affect the recording or ascertainment of endpoints. Which brings me to the next element, well-defined and reliable assessments of subject responses. This is basically getting good endpoints. And you already heard a talk on cardiovascular endpoints, but this is a very important part. We need good measurement techniques that produce, clinically relevant data. And finally, the last element is adequate analysis to assess drug results. This is where the number crunching comes in, where most people think statistics has a primary role. But as I said previously, statistics has a role throughout these elements. Okay, related to assigning patients to treatment and control groups, I'd like to cover the difference between a randomized study and an observational study. In a randomized study, let's say a patient comes along and there's some random process that assigns the patient to the two treatment arms, whether they receive drug A or drug B. Here, I show it as a flipping of a coin, heads or tails. And in practice, of course, that would be likely computer generated. In an observational study, a patient comes along and the study is not intervening in what the patient gets. That's done through typical healthcare practices. So the healthcare provider and the patient basically determine what drug the patient may end up on. And the healthcare provider may use lab tests or demographics like age, sex, race. They may likely use medical and family history, concomitant drugs, even what insurance they get or what's convenient for the patient in terms of administering the drug. And there might be differences in different geographical regions. So you can see there's a lot of information that goes into the choice of the drug. And this is done in hopefully the best interest of the patient. So where does that leave us? So in a randomized study, the characteristics of patients receiving drug A are similar to the characteristics of patients receiving drug B. It was just a random flip of a coin where you ended up, drug A or drug B, being male or being over 65 or having a particular medical history doesn't affect whether you go to drug A or drug B. However, in an observational study, the characteristics of patients receiving drug A may not be similar to the characteristics of patients receiving drug B. As I said, there's in medical practice, it's the actual characteristics that may determine what drug you get. So this brings me to confounding. Without randomization, there may be systematic differences or biases when comparing people getting drug A and people getting drug B. This is known as confounding. So, for example, suppose drug A is given to older, sicker people. Perhaps it has less side effects and is easier to tolerate. So even if there was no differences in the actual effect of drug A and drug B, if we did an observational study without consideration of confounding, the comparison may show that drug A has worse outcomes. This is because the people getting drug A are starting off worse. So again, this is something that has to be considered in an observational study, and sometimes it makes an observational study not practical to achieve its objectives. I talk about confounding here, but that's just one of many sources of bias. There are other sources of bias to be concerned about, even with randomization. We already talked a little bit about blinding, so that's bias on the part of observers. So if the patient or healthcare provider know what drug someone's on, that may actually bias the results, and I think you're all aware of that. I would hazard to say that many of the studies you hear about in the news or the ones that make big headlines, at least outside of medical journals, are observational studies. And although observational studies can be done very well in certain settings, they're often not done well. They're very kind of inexpensive and potentially easy to do. So I would say, take them with a grain of salt. As a rule of thumb, anything you hear about coffee, red wine, or chocolate is probably not based on strong evidence. Now I would like to talk about variability versus bias. Here we have 4 targets in which the center is a true drug effect. The upper left is the best situation, low variability and low bias. The points are close together and they are centered on the truth. Moving to the top right, we have high variability and low bias. The points are spread apart but are centered on the truth. In the lower left, we have low variability and high bias. The points are close together but are not centered on the truth. Finally, in the lower right, we have the worst case of high variability and bias. Well, if only it was that easy. In practice, of course, we don't have the targets because we don't know what the true drug effect is, so now we wouldn't be doing the study. All we see are the data. So taking a look at the left-hand part of this figure, the top and the bottom, we see a close grouping of points, so we see low variability. But without the target, we don't know anything about the bias. We don't know if the top is biased or the bottom is biased, or perhaps they're both biased. Likewise, on the right-hand side of this figure, we see a larger scattering of data, more variability, but we don't know if the top or the bottom or both are biased. So bias is sort of our unknown unknown, whereas variability is sort of our known unknown. We see variability, we don't see bias, and that's why it's important to address bias the best you can at every stage, the design, the conduct, and the analysis. So let me reiterate a few points here. Statistics can help quantify variability. As I said, we see variability, we're able to quantify it. And this is done in the analysis stage. Typically, and you produce things like confidence intervals and p-values. Design and conduct are generally where you reduce bias. And again, that's important because you don't see bias, so you have to do everything you can to prevent it. And we saw some methods to reduce bias, including randomization and blinding. And of course, good conduct is important overall to reduce bias. And I've made this point already, and I'll make it again a few times throughout the talk, that statistics can help reduce bias and variability at each stage, the design, the conduct, and analysis stages. Well, there are multiple ways of reducing variability in a study. I mean, for example, you can have a better measurement process for endpoints. But perhaps the most intuitive and straightforward is increasing the sample size. So of course, sample size is the number of people in the study. I think we all have some intuition here. So what is a better estimate of the average age of this session's attendees? Well, we could pick a random sample of 5 attendees and calculate their age. Or we could pick a larger random sample of 20 attendees and calculate their age. I think we all sort of intuitively understand with more people in our sample, we're sort of averaging out variation. And the larger sample will have lower variation. Now I'm going to switch gears to some other basic statistical concepts. I'm going to talk about hypothesis testing. Hypothesis testing is somewhat tricky. When I first was exposed to it as an undergraduate, I found it very confusing. It's not mathematically tricky. It's tricky because of the terminology and the logic. So you may at some point have to go back and review these slides if you feel that's helpful to sort of understand the logic and the terminology a little better, but I'm going to go through it. So the null hypothesis is typically what you're trying to show is not true. So for example, drug A and placebo have the same effect or no effect. That's what you're trying to show is not true. The alternative hypothesis, that's typically what you're trying to show. So following the example, drug A has a better effect than placebo. I'm going to carry this sort of example along that the null hypothesis is no effect and the alternative hypothesis is an effect. But that's not always the hypotheses you're going to have. I won't go into it in any detail, but some of you may have heard of non-inferiority trials where you're trying to show that their two drugs are the same, or at least non-inferior to each other. So in that case, the role of the null and the alternative swap. But I'm going to stick with this example. But whatever case you're in, the null is what you're trying to show is not true, and the alternative is what you're trying to show is true. And so again, the terminology is perhaps a little confusing. Okay, it's actually going to get a little worse with p-values. P-values are the probability of observing an effect or something more extreme if the null hypothesis is true. That statement, if the null hypothesis is true, is important here. So for example, if we're doing a study of blood pressure, the study might estimate a drug effect of a reduction of 5 millimeters of diastolic pressure. So that we have observed this 5, so the p-value is the probability of observing 5 or even something more extreme, 6, 7, 8, but under the assumption that the drug had no effect. So small p-values are evidence against the null hypothesis, or following my example through, evidence that there is no drug effects, so therefore there's probably a drug effect. So suppose we got a p-value of 0.02. If there was no drug effect, the chances of seeing this, what we saw, or something more extreme, is only 0.02. This is small. It leads us to doubt that the null hypothesis is true. It leads us to doubt that there is no drug effect. So we might conclude that there is a drug effect. So just to note, the p-value is not the probability that the null is true. This is important to keep in mind. I'm not covering confidence intervals, but sometimes, confidence intervals are not probability. But if you mix them up, you're kind of not in the worst shape. But it's, I think, important to keep in mind that p-values are not the probability that the null is true. It's this more complicated logic that what you observe is very unlikely under the null. Okay, now I'm going to say a few words on decision errors. So in hypothesis testing, at the end you either conclude a drug works or there's not evidence that the drug works. And you could be right. We don't know what the true answer is, but you can be right that you concluded the drug works when it actually does. But you can also make some errors, and there are basically two types of errors you can make. You can conclude that the drug has an effect when it does not. This is known as a type 1 error. And to a large extent, this is FDA's and society's problem. We're putting a drug on the market that doesn't have an effect. But it's also, of course, the drug company's problem as well. They don't want to put a drug on the market that doesn't have an effect. The other type of error is sort of the reverse of that. We conclude that the drug does not have an effect when it actually does. This is a type 2 error. Of course, this is the drug company's problem. They have an effective drug, but they don't have evidence that it works. But it's, of course, FDA's and society's problem as well, because we all want effective and safe drugs on the market. Okay, let me describe how we use p-values for hypothesis testing. Remember, p-values are the probability of some extreme observation under the null hypothesis. So we reject the null hypothesis if the p-value is below some value alpha. So alpha is sometimes called the significance level. And this is equivalent to saying the drug has an effect, using the example I've been using. And we typically use small alphas like 0.5. So there's only, if the drug has no effect, we only have a 5% chance of rejecting the null hypothesis and concluding the drug has an effect. So we're really limiting what I call type 1 error on the previous slide. So we typically use these small alphas. Okay, let me try to put this all together here and how this is done in practice. We can set the type 1 error by choosing alpha. So again, we typically choose an alpha of 0.05. So we have a 5% chance of making a type 1 error, a 5% chance of rejecting the null when the null is actually true. We can limit the type 2 error by having larger sample size or more patients. We already spoke about that, more sample size is equal to less variability, and what this will make it less likely to conclude the drug has no effect when it really does. So that will reduce the type 2 error. We typically have type 2 errors of 10 or 20%, and that's typically expressed in terms of power. So we have 80 or 90% probability to reject the null when the alternative is true. Okay, now I'm going to move on to another topic, multiplicity. I think this topic will be a little easier to comprehend than the previous topic. Multiplicity is also known as multiple bites from the apple. So I have a toy example to try to kind of display this concept. Okay, suppose we're trying to determine if a drug has an effect, but the drug we're looking at has no effect. So it's basically a coin toss whether you have a good outcome or a bad outcome. So we're going to simulate the study by flipping a coin 4 times, and every time you get an H, that's a good outcome for a patient, and a T is a bad outcome for a patient. Our decision rule is if we get 1, 2, or 3 heads, we conclude the drug has no effect. If we get 4 heads, we conclude the drug has an effect. So we do the study 1, and it looks like we get H, H, T, H. So by our decision rule, we conclude the drug has no effect. But we're not happy with that result, so we do the study again. And in study 2, we get H, T, T, T. And again, we conclude the drug has no effect. So we do it again, and study 3 also concludes that the drug has no effect. So eventually, after we do this 12 times, we get 4 H's, and we conclude the drug has an effect, and we publish a paper on it. Now, I think you can all understand the problem with this. If you do enough studies, or if you look at the data in many ways, you will see an effect, even when there is no effect. This is known as data dredging or p-value hacking. It is a known problem in science. Okay, well, multiplicity can show up in many ways, and it chiefly shows up by looking at multiple subgroups or multiple endpoints. So multiple subgroups might be, in your study, you look at the effect on males, the effect on females, the effect on people over 65. And if you look at enough subgroups, you're likely going to see some effect. But whether that's random chance or something real, it's unclear. So this sort of thing is more, looking at multiple subgroups like this is often hypothesis generating and not confirmatory. Similarly, you can look at multiple endpoints, the effect on blood pressure, the effect on life expectancy, the effect on happiness. Again, if you look at enough, you're bound to find something, and you don't know whether that's random chance or something real. So again, it's more hypothesis generating than anything confirmatory. And the way to sort of address multiplicity, so when you do find something, you could be more clear that it's a true finding, is through pre-specification. You tell the world ahead of time what you primarily look at, what endpoints of interest, what populations of interest, and that's typically specified in the protocol and statistical analysis plan. There are also statistical methods to address multiplicity that I won't get into today. Okay, now on to the challenge questions. The first question is, which of the following does not reduce bias? A larger sample size, randomization, blinding the knowledge of the drug from study participants and investigators, or pre-specification in the protocol and statistical analysis plan. I'll just pause a second for you to think about this. Okay, the answer is a larger sample size. Larger sample sizes will reduce variability, but may not affect bias. As we saw in those bullseye examples, you can have low variability, but still bias. Okay, the next challenge question, the answers are the same, but the question is different. Which of the following addresses multiplicity? A larger sample size, randomization, blinding the knowledge of the drug, or pre-specification in the protocol and statistical analysis plan? Okay, here the answer is D. We discussed that pre-specification in the protocol and the statistical analysis plan are primarily how you address multiplicity. That, as well as some statistical approaches that would be specified in the statistical analysis plan. Here are some resources for further reading if you're interested. Unfortunately, I won't have time to go into details of each of them. I would start on the top and work my way down to the bottom. They go from general to more specific. And the overarching statistical guidance is that ICH E9 statistical principles for clinical trials. Okay, well, thank you for your attention, and I'll be taking questions at the end of this session. Thank you.
Thank you, Dr. Sachs, for that introduction. So good afternoon, everyone. My name is Shabnam Naseer, and today I'll be talking about safety considerations in clinical drug development. So here are some of our learning objectives. One, we want to understand that protection of subjects should always be the first priority when designing early clinical studies. We also would like to learn a few key safety considerations in the conduct of phase 1 trials, and also to understand the principles of safety monitoring and reporting in clinical trials. The main objectives of phase 1 trials are the following: to assess safety and tolerability of the drug product, to attempt to characterize dose-limiting adverse reactions, to determine maximum dose associated with an acceptable safety profile, to characterize pharmacokinetic parameters, and explore drug metabolism and drug interactions. The subjects in phase 1 trials generally tend to be healthy volunteers who have less confounding factors. In some instances, patients are enrolled into phase 1 trials when the drug is known or expected to be toxic, and an example of that is with cytotoxic agents. When patients are enrolled in phase 1 trials, there will be more confounding factors and sometimes more difficulty in separating disease-related manifestations from adverse reactions. Special populations such as those with renal or hepatic impairment can also be enrolled into phase 1 trials. So here are some general considerations when one is moving from the non-clinical to first-in-human studies. We have to consider what is the evidence we have garnered so far from the non-clinical studies. And some of the questions we'd like to ask are, what are the duration and total exposure proposed in humans, and how does that correlate with what was tested in the animals? What are the characteristics of the test drug? Is it a small molecule, a biologic, or does it have a long half-life? What is the disease being targeted for treatment? Because that would be taken into consideration in the benefit-risk assessment. What are the populations in which the drug will be used? Will women of childbearing potential be enrolled in these studies, or will studies be conducted in children? Is the product being administered systemically or topically? Someone has to think about all of these factors. Some other considerations include, do the non-clinical studies provide sufficient safety support for the proposed clinical studies? So what are the species that they are studied in? Are these species appropriate, and are they relevant to the particular drug? Were the potential target organs of toxicity identified? What was the duration studied in the animals? Was the dose and route of exposure appropriate and similar to what is being proposed in the human studies? Were the PK and PD assessments appropriate? Was the dose response identified? And again, do we need to do any special studies in animals if one is planning to enroll children or pregnant women? So it's important to keep in mind that only some toxicities that are seen in the non-clinical studies translate into adverse reactions in humans. But there are other toxicities that do not. And so you end up with both predictable and unpredictable toxicities, and these can appear in any phase of development, and sometimes you only see them post-marketing. It really depends on the size of your safety database that is available pre-market. Also, you want to keep in mind that certain adverse reactions that are subjective, such as a headache, or other events such as hypersensitivity reactions, cannot really be assessed in non-clinical testing. So here's an example of a predictable toxicity from the anti-infective world, and I'm sure you can find similar examples in other therapeutic areas as well. So just some background, linezolid is an antibacterial drug approved in the late 1990s, and at that time it was the first member of its class in the oxalidenone class. It had activity against gram-positive organisms, including some resistant organisms, so there was some therapeutic benefit that could be offered by this product at that time. The non-clinical studies that had been conducted showed that myelosuppression occurred, and this adverse event was both dose and time dependent. There were various changes in the bone marrow, and it affected all 3 of the cell lines. But the product had the potential to offer a therapeutic benefit, and further clinical development was pursued. When phase 3 trials were conducted, there was no evidence of myelosuppression per se, but there was a high frequency of thrombocytopenia seen in the linezolid-treated patients. At the time the product was approved, the prescribing information included precautions for thrombocytopenia, and the animal pharmacology section described the hematopoietic effects noted in animals. After the product was approved, there were cases of myelosuppression affecting all 3 cell lines that were reported. So then the labeling was updated to include a warning regarding myelosuppression. So on the other hand, we'll now talk about an example of unpredictable toxicity. And again, this is also from the anti-infective world, since I have most experience with that. So we had 2 products, both were members of the beta-lactam class, which is a very well-known class of antibacterial drugs, for which we have a lot of experience in humans. The structure of these beta-lactams, however, was modified to enhance the spectrum of activity. There were no unexpected toxicities seen in animals, so both of the sponsors proceeded to conduct their phase 1 trials. The single-dose phase went well, but then when they went into the multiple-dose trials, the subjects developed moderate to severe skin reactions, such that the product development for both products had to be halted. So even though the non-clinical studies appear to be pretty benign and you don't see any adverse events, it doesn't mean that you wouldn't see it when you went into humans. So now we'll move on to the concept of maximum recommended starting dose. There are key principles in selecting an MRSD. One is that you want to avoid toxicity at the initial clinical dose, but at the same time you want to allow reasonably rapid attainment of the trial objectives, usually tolerability and PK, so that you're not that low that it will take you forever to get to where your targeted dose was. Typically, we use an algorithmic approach based on what doses were given to animals and what toxicities were observed. There are some alternate approaches based on animal pharmacokinetics and modeling, but for the most part we use the algorithmic approach. So some of the key concepts when you look at the MRSD are the no observed adverse effects level, or NOEL, and that's the highest dose tested in animal species that did not produce a significant increase in adverse effects compared to a control group. Then the second term is human equivalent dose, or HED, which is really what is the dose in the animals that converts in a certain milligram per kilogram dose in humans. Based on that, you would be able to come up with a starting dose. Then you would have to consider which animal species you use to make these calculations. Ideally, you would choose the most sensitive species, which is the species from which you can identify the lowest HED. In some cases, you would have to use the most appropriate animal species because that species might have the particular target of interest, and this is especially seen with biologics. So here's a schematic showing the steps in determining the maximum recommended dose. First, you would determine the NOEL, which is a starting point for determining a safe starting dose for new molecular entities in healthy volunteers. Then you would convert that into the HED. You select the HED from the most sensitive or most appropriate species, and then you choose a safety factor. Then you divide the HED by that safety factor to come up with a maximum recommended starting dose. So what is the safety factor? This is a number which gives you a margin of safety for protection of human subjects when you're moving from animal studies to humans receiving the initial clinical dose. The default number is 10, but there is some degree of flexibility. Sometimes we can go higher or lower. The safety factor provides us some reassurance that when moving from animals to humans, that you're not exposing human subjects to any great risk. One knows that there is variability when you move from animals to humans, and the safety factor allows for variability to be accounted for. So, as mentioned previously in some of the examples, there are uncertainties when you go from animals to humans. There can be differences in the sensitivity between humans and animals to certain products, difficulty in detecting certain toxicities in animals, differences in receptor densities. You always have to be prepared for unexpected toxicities and interspecies differences in how the product is handled. Animals and humans may not always handle the product in an identical manner. When would you have to increase safety factor greater than 10? If it's a brand new class we have no prior experience with the product is one reason. The toxicities in the non-clinical studies were severe or irreversible, or they were non-monitorable. For instance, if you have histopathologic changes in the animals, but they were not monitored because there are no clinical methods to monitor the toxicities, or there are no biomarkers. You may want to increase the safety factor if there's a steep dose response curve, if the pharmacokinetics are not linear, or if there's variable bioavailability because you may have poor bioavailability in the animals that may actually underestimate the toxicity when you move to humans. So on the other hand, you may be able to decrease the safety factor to less than 10 if you're dealing with a member of a well-characterized class of drugs, or if the toxicities that are produced are easily monitored, reversible, or predictable, or if the NOEL was determined based on toxicity studies of longer duration, you might have a bit more reassurance in reducing the safety factor. So let's discuss an example of a calculation of MRSD. Here's a product for which the HED was about 400 milligrams. The sponsor proposed a starting dose of 100 milligrams, so that brings the safety factor to 4. The rationale provided was that this was a member of a well-characterized class of drugs, so using a factor of less than 10 was okay. Also, they noted that toxicity studies in 2 species were of longer duration than what was proposed in the clinical trials, and that these toxicities were readily monitorable and reversible. So we did not agree with this rationale because we knew that members of the class had exhibited more toxicity than the parent class from which it was derived, and we were also concerned that the bioavailability in animals was low and that human bioavailability could be greater, leading to a risk of higher exposure in humans. So we agreed upon a starting dose of 50 milligrams, so the safety factor was increased to 8. So now let's move into some general considerations when you're conducting these phase 1 trials. Some of the questions you have to ask yourself when you're getting ready to do these trials are the following. Are the clinical trial protocols designed appropriately to ensure safety and meet the stated objectives? You have to protect the safety of the subjects in the trial. Do you have enough information about the quality of the investigational products? Has the formulation been well characterized, and do we know what the impurities are? Are the route and the rate of administration appropriate? Can the product be given as a bolus dose, or are the toxicities such that it has to be given as a slow infusion? What is the mechanism of action? Is it novel? Do we know anything about similar products that act via a similar mechanism? Are you expecting an off-target effect? And that's very important, especially if you're dealing with a biologic and the mode of action involves a target which is connected to multiple signaling pathways or affects biologic cascade or cytokine release. So how do we determine an appropriate dose? Ideally, one subject should receive a single dose, and then that is followed by sequential administration within each cohort. You want to ensure that there's an adequate period of observation between dosing so that one has the time to observe and interpret adverse reactions in the cohorts at the lower dose. How long you observe will depend again on the properties of the drugs, What are their PK properties? How long does the drug stay in the system? How long is its half-life? And we also learn good lessons from previous trials conducted with similar products. So if the protocol for these phase 1 trials allows for dose escalation, you have to ask whether the dose escalation is appropriate. Are the dose increments appropriate? Is it okay to double it or go up by 4 times? You have to be cautious and use a slower rate of dose escalation if you have a small therapeutic window, or if your animal models were not very good, or if you have any concerns about toxicity. There's a bit of flexibility here, and again, it depends on your specific product, on your target, and toxicities seen in the animal studies. You also have to ask yourself, is the amount of information before each dose escalation appropriate, and are the number of subjects at each dose appropriate? So it's important that the protocols have an appropriate monitoring scheme to look at clinical signs and symptoms of adverse events likely to be associated with the drug. The duration of clinical observation should be adequate with respect to the stated objectives and endpoints, the anticipated response to the product, and health-related conditions being studied. Sometimes we do need to monitor for longer periods of time, and again, it all depends on the characteristics of the drug. If you're worried about any sort of delayed onset reactions, unexpected toxicities, you have to make sure that the follow-up is going to be long enough. So as far as the frequency of monitoring, there may be a need to observe more frequently soon after initial dosing in the first week or so, and certainly you would need follow-up visits for subjects who develop adverse events or laboratory abnormalities. In addition to monitoring clinically for adverse reaction, you would also need to monitor laboratory tests, and which particular test you order and collect the data on, again, really depends on the drug, depends on the target organs of toxicity. You want to consider whether routine assessment of all organ systems in the laboratory tests is needed. Do we know enough about the drug such that you only need to assess specific organ systems? Are the laboratory tests sufficiently detailed so that you can make an assessment of whether or not the drug is causing toxicity of those specific organs? And lastly, are there stopping rules based on laboratory abnormalities if they reach a certain threshold? So we talked a little bit about stopping rules on the previous slide. So what are stopping rules? It's important that the protocol has clear stopping rules for administering the drug, enrollment, stopping enrollment, stopping dose escalation, and rules for termination of the trial. Sometimes we've had to put INDs on hold because there were no stopping rules in the protocol. So again, these are changes to the protocol that are implemented when toxicity is observed. And to be able to have stopping rules, you have to identify what might be acceptable toxicities and what might not be acceptable toxicities. Since it's unlikely that that you'll have a drug with no toxicities, it's important to try to generate these stopping rules. So if acceptable toxicities occur, these will not result in changes in how the trial is continued in terms of subject enrollment and dosing. If you have other toxicities that are not acceptable, you have to have a procedure in place for what you would do with the study. So that brings us to our first challenge question. So which of the following is used to calculate the maximum recommended starting dose, or MRSD? So the answer here is A, which is the Noel, and we talked about this on several slides previously. So I hope you got that one right. Okay, so now we've talked a lot about safety and phase 1 trials, but the evaluation of safety is actually an evolving process, and it depends on the stage of the product under development. For products that are marketed, the safety information is reflected in the product labeling, also known as prescribing information or PI, while up-to-date safety information for the products under investigation, including the non-clinical findings from the in vitro and animal studies, clinical, pharmacologic, and safety data, when available, is found in the investigator's brochure or IB. For unapproved products, the investigator's brochure is equivalent to the prescribing information. So what are the sources of safety information found in the investigator brochure or prescribing information? So you can have non-clinical data, including in vitro and animal data, as well as information about the structure of the product and the manufacturing process. Clinical pharmacologic data can include information about PK/PD. Safety information can come from clinical safety data in healthy volunteers and patients, as we just discussed. But the most pertinent information, of course, will be the data from the clinical trial for the same indication, as such data is generally controlled for the patient population characteristics and underlying pathologic process, and it can also come from trials for different indications. Post-marketing experience, the published medical literature, and last but not least, safety profiles of other drugs in the same class can give you safety information. So again, why is safety monitoring required in all clinical trials? So essentially the goal of safety monitoring, as we had discussed, is to identify, evaluate, minimize, and appropriately manage risk. And this will lead into our discussion of adverse event ascertainment and monitoring. So we do have a couple of regulatory definitions that we want to discuss. What do we consider an adverse event? And this is in the Code of Federal Regulations, and I have provided the CFRs in the slides. So an adverse event is any untoward medical occurrence associated with the use of a drug in humans, whether or not it is considered to be drug-related. It could be a sign, a symptom, or a disease, an abnormal laboratory finding, vital sign, imaging finding, or other testing, worsening, or a constellation of the above. An adverse event can also be referred to as an adverse experience. So some examples of ways that adverse events can be ascertained include spontaneously reported or observed signs or symptoms, or symptoms or signs that are reported as a result of a probe, such as a questionnaire or a checklist, testing of vital signs, laboratory data, or special safety assessments, which can include, for example, visual or hearing assessments. So upon reporting, adverse events are characterized according to their severity. There are a number of severity grading scales or tables available to provide guidance to the sponsors or the investigators for safety monitoring in clinical trials. And again, the use of these systems is optional. They are specific to the study population. For example, whether you're studying patients or healthy volunteers, clinical abnormalities may be presented as, for example, mild, moderate, severe, or potentially life-threatening in healthy volunteers. And in oncology patients, the AEs can be graded by a scale of 1 through 5, where 1 is mild symptoms and grade 5 is death related to the AE. So it's important to keep in mind that the different scales are specific to these different characteristics we just spoke about. And the term severe is also not the same as serious in the classification of AEs. I also have some examples of AE severity grading scales listed there. So this brings us to the second regulatory definition, which is serious adverse event. And again, serious is not to be confused with severe. Serious adverse event is any adverse event that, in the opinion of the investigator or the sponsor, results in death or is life-threatening, results in hospitalization or prolongation of an existing hospitalization, a disability, birth defect, congenital anomaly, or an important medical event. Those are basically ones that may jeopardize the patient or require intervention to prevent one of the outcomes listed above. An example of this is allergic bronchospasm, which required intensive treatment in an ER or at home. So the investigator's perspective here is important because it may have been informed by actually observing the event, while the sponsor is likely to have broader knowledge of the drug and its effects to inform its evaluation of the significance of the event. Here are some examples of uncommon SAEs that should raise a flag that there is a possibility that the AE was caused by the drug, by the study drug, especially in a study population of generally healthy individuals. So this is just meant to be examples. It's not a comprehensive list, but perhaps a good place to start. Serious adverse events, like any other adverse events, need to be assessed for drug-related causality, and a number of questions may help in that assessment. So one could be, is it a common occurrence in the population under study? Was it treatment emergent, meaning did it occur on treatment or shortly thereafter? Did it respond to drug discontinuation or de-challenge? Did it occur upon reintroduction of the study drug or re-challenge? Were there concomitant medications? Were there laboratory or other tests done to characterize the adverse event further? Was there an obvious alternative cause? And is the SAE a study endpoint? For example, was a death also a study endpoint? Please take a note that when reporting serious adverse events, you should try to provide enough relevant clinical information in the case report form to allow for good quality narratives. So now we're going to talk about AE reporting requirements, what the investigator would report to the sponsor, and I have the CFR references there on the slide. So again, what does the clinical investigator report to the sponsor? An investigator must immediately report all SAEs, whether or not the event is considered drug-related. The investigator must also include an assessment of whether there is a reasonable possibility that the drug caused the event. Study endpoints that are SAEs must be reported in accordance to the protocol unless there is evidence suggesting a causal relationship between the drug and the event. In that case, the investigator must immediately report the SAE to the sponsor. The investigator must record non-serious AEs as well and report them to the sponsor according to the timetable in the protocol. So let's discuss a hypothetical case, case one, in which you're the investigator for a clinical trial evaluating whether antihypertensive drug A is associated with a reduced risk of death, MI, or stroke, and a 75-year-old white male has just died in the clinical trial. So the question is, do you have to immediately report this case to the sponsor? I'll let you think for a few minutes. So basically, we don't have enough information on the slide to determine causality. However, by definition, this is an SAE because the patient died, and death is also a study endpoint in this trial, as you can see that the drug A is being evaluated to determine whether there's a reduced risk of death or other events. So the investigator must report this event in accordance with the protocol, since it's a study endpoint, that's an SAE, unless there's evidence indicating that the SAE is causally related to the drug. So if I tell you that the death in this 75-year-old male was due to anaphylaxis, that will sort of change things, and now there's evidence suggesting a causal relationship between the drug and the event. So you, as the clinical investigator, must immediately report the event to the sponsor, even if the event is a study endpoint. So let's talk a little bit about what happens when an AE report gets to the sponsor. So basically, they're converted from the investigator's verbatim terms to the standardized preferred terms using a specialized dictionary called MEDRA. And MEDRA is the medical dictionary for regulatory activities, which can be used to sort AEs and group like events. Preferred terms are used to calculate the incidence of AE. And this is just a schematic showing the structural hierarchy of MEDRA. The highest level of terminology and least specific is the SOC, or the system organ class, and then it breaks down all the way to the lowest level term, which is also referred to as the verbatim term, and that's the AE reported on the case report forms. And again, that's converted to PT, which represents a single specific medical concept, or again, preferred term. So clinical investigators do play a crucial role in safety reporting using the most scientific and consistent terminology when reporting adverse events, as coding problems might result in splitting or lumping adverse events and therefore missing safety signals during the safety data analysis. So splitting similar terms such as hypertension and high blood pressure can result in a lower incidence or may minimize or mask a safety signal. Lumping dissimilar terms to the same PT may obscure a safety signal under the lump term. For example, facial edema is very different pathophysiologically than leg edema, for example, and a coding issue could be lumping all edema to the PT edema. In the third category, there may be a lack of an adequate term or definition to represent a syndrome of interest, such as serotonin syndrome, and in this case, you may need a prospective case definition if the syndrome is not well characterized by a single term. So moving back to regulatory definitions, now the ones that are important to the expedited reporting of adverse events to the FDA. The concept of unexpected adverse event is presented here, and this is an adverse event. An adverse event is deemed unexpected if the following conditions are met. So one is that it's not listed in the investigator brochure, or if the IB is not available or required. If it's listed in the IB but not at the specificity or severity observed, and one example of this is the IB lists increased creatinine, but you actually have a case of acute renal failure, which would be unexpected by virtue of greater severity. Also, it would be an adverse event would be unexpected if it's not consistent with the risk information described in the general investigational plan or elsewhere in the current application, or if it's mentioned in the IB as anticipated due to pharmacologic properties of the drug, or if it occurred with other drugs in the class, but not with the particular drug under investigation. So, for example, angioedema was anticipated to occur in some patients exposed to a drug in the ACE inhibitor class, and angioedema would be described in the IB as a class effect, but a case of angioedema observed with the drug under investigation should be considered unexpected for reporting purposes until it is included in the IB as occurring with the drug under investigation. So now we have our hypothetical case 2. You're in the investigator for a clinical trial evaluating a new quinolone antibacterial drug B for the treatment of pneumonia. The investigator brochure lists a number of SAEs associated with the use of quinolone drugs, including neurotoxicity. And so the question is a seizure in this trial considered an expected AE? So sorry I advanced the slide, but going back to our definition of unexpectedness, we know that this is a class adverse event, and neurotoxicity is a pretty broad event, but let's say that seizure is in the investigational brochure as a class-related event, and is clearly noted in the prescribing information for quinolone drugs as an adverse event for this class of drugs as a class effect. But however, at the moment, the investigator's brochure does not report that this adverse event had occurred for the drug under study. So seizure in this hypothetical case too would be considered unexpected. So this is our third regulatory definition. It's suspected adverse reaction. And so a suspected adverse reaction is an adverse event for which there is a reasonable possibility that the drug caused the AE. And again, reasonable possibility means that there's evidence to suggest a causal relationship between the drug and the adverse event for the purposes of IND safety reporting. Some examples are listed here. A single occurrence of an uncommon event that is known to be strongly associated with drug exposure. So some examples of this would be Stevens-Johnson syndrome or angioedema. If you have one or more occurrences of an event not commonly associated with drug exposure, but otherwise uncommon in the exposed population, for example, neutropenia in healthy subjects. Or another example is an aggregate analysis of specific events observed in a clinical trial that indicate that those events occur more frequently in the drug treatment group than in a concurrent or historical control group. An example of this could be acute myocardial infarction in a long duration trial with elderly cancer patients. So the determination of an AE as a suspected adverse reaction, it is difficult but critical to avoid submission of uninformative IND safety reports. The sponsor should evaluate the available information and decide whether there's a reasonable possibility that the drug caused the AE, as the sponsor is the ultimate party that determines if the AE is or is not causally related to the AE. So this slide illustrates the universe of suspected adverse reactions, which are no longer just adverse events with undetermined causality that are noted in the outermost sphere, but not yet a definitively established adverse reaction, which are noted in the dark red sphere in the center. Adverse reactions are a subset of all suspected adverse reactions where there is a reason to conclude that the drug caused the event. So again, suspected adverse reactions are those AEs where there's evidence to suggest a causal relationship between the drug and the AE. So FDA continues to require that 3 criteria generally be met before a sponsor submits an IND safety report. So, namely, the event must be serious, unexpected, and there must be evidence to suggest a causal relationship between the drug and the event. So often this standard is referred to as a SUSAR. And the sponsor must assess whether or not there is a reasonable possibility that the drug caused the event. The sponsor should only submit these type of IND safety reports where all 3 criteria are met. And in addition, just to clarify, sponsors are also expected to submit an annual IND report that includes a summary of the most frequent adverse events, which are not reported individually, in addition to summarizing the SAEs. Sponsors have 2 timeframes when it comes to IND safety reports. Those reports that are required to be submitted to FDA in 15 days and those required to be submitted within 7 days. As you can imagine, the most serious events must be reported within 7 days. So there are 5 categories of adverse events that trigger the sponsor's responsibility to submit an IND safety report, namely SUSAR, which we just discussed, and then also other criteria as noted here, and the CFRs are listed there for your reference as well. So sponsors have to submit IND safety report, again, within 15 days of the adverse reaction, unless the adverse reaction was fatal or life-threatening. And in this case, the report must be submitted to FDA within 7 days. So we have our last case, hypothetical case 3. So in a trial of a marketed HIV drug C, an 8-month-old infant enrolled at 1 month of age was noted at study month 4 to have moderate hearing loss in a clinic progress note. So the question is, should this event have been reported to the sponsor by the investigator immediately? So we have to think back to our definition of a serious adverse event. In this case, hearing loss is a disability, and thus it is an SAE. All SAEs, regardless of causality, should be reported to the sponsor by an investigator. So the answer here is yes. In addition, the investigator should immediately report the SAE to the sponsor and also submit a assessment of causality. So we've actually come to the end of this lengthy talk. In summary, I've provided a high-level overview of safety considerations in phase 1 trials and some important questions to ask yourself before you decide to dose humans as you translate the information from non-clinical studies to either healthy volunteers or patients. What is the relevance of those toxicities seen in the non-clinical studies and how they translate to adverse events in humans? It's important to keep in mind that there are both predictable and unpredictable toxicities. We discussed how to determine a safe starting dose in humans and the concept of the safety factor. We discussed the importance of safety monitoring and stopping rules. We moved on to the ascertainment of safety in larger clinical trials and important regulatory definitions of adverse events. Finally, we discussed how the investigators play an integral role in assuring quality safety assessment by reporting to the sponsor, and then in turn how the sponsor will evaluate all available safety information and report to the FDA and all the participating investigators as well. So challenge question 2. This is a true or false. An investigator would not have to report a case of hepatic failure if the investigator brochure listed elevated hepatic enzymes or hepatitis. So the answer here is false, because the case of hepatic failure is at a much greater severity than what is listed in the investigator brochure. So this is false. Okay, so thank you so much. These are my references, 2 pages of references for you to peruse. And once again, thank you so much for giving me the opportunity and for your attention today.
Well, thank you very much, Mark and Shabnam, for very comprehensive talks. As we get our cameras on, I'm going to scroll through some of the first questions in the chat. I see we've got a whole lot of them. Let's see. Maybe we can start off with a question for Mark. Mark, let's just see if you're on.
I am on.
Great. And the question I see here for you is, Does your presentation equally apply to device studies, or are there any variations? Do you want to take that one?
Sure, yes. As we were reviewing the questions, I noticed a number of questions on devices. I would say all the principles I discussed in my talk do apply to devices. I think the one area maybe just to be a little careful about is on one slide I presented regulations, adequate and well-controlled study. And those regulations are, I believe there are similar regulations for devices, but I'm not entirely sure about that. But regardless, I think the principles in what makes an adequate and well-controlled study apply to devices and even in non-regulatory settings.
Great. And now I think let's turn to Shabnam. Yeah, you're on. So I see there are quite a number of questions about safety. Here's one which I have in front of me. Who's primarily responsible for investigating further adverse events or severe adverse event issues? Is it the sponsor or FDA? I think that's an important question because sometimes the data we get is not sufficient on its face. Any thoughts there, Shabnam?
Yeah, thank you for the question. I think the assessment of safety and reporting safety is it's a role that's shared by the investigator and the sponsor, and then ultimately FDA as well if the IND safety report is reported to FDA. So for investigators, basically the role of the investigator is to determine if the AE is serious, and if they feel that it's serious, they would immediately report at the sponsor. The investigator should also include a causality assessment for each of the SAEs that are reported to the sponsor because, they have contact with the patient, they have firsthand knowledge, so they can make that causality assessment and include that when they report to the sponsor. And they would also want to review IND safety reports and report any unanticipated problems involving risk to human subjects. Where the sponsor has knowledge of not only that investigational drug, but perhaps other clinical trials in which that a drug may have been evaluated, or drugs in a similar class. So they have a broader scope, and they can evaluate both individual and aggregate safety signals. They also, like the investigator, determine if the SAE is causally related to the investigational drug, and they're primarily responsible for that role to determine causality. And then the sponsor report the events meeting criteria, as we had discussed, meeting criteria as IND and to all participating investigators.
Great, I think.
And then, like I said, when the report comes to FDA, the team that's involved with that investigational drug would also review the safety report.
Yeah, I think that's also an important aspect. I mean, I don't know if you've had that experience as well, Shabnam, but maybe, when a reviewer goes through a report, there may be additional information that's needed. And I think we would follow up quite rigorously about that if the event was really important to the approval.
Yeah, correct, Leonard. We can, ask for more information with the sponsor if it's necessary, narratives, summaries, and things like that, if we need to make a determination.
A different sort of question for Mark now. Which is one close to my heart. Can you discuss how FDA addresses the vulnerabilities on non-inferiority trial designs? And the examples given here are assay insensitivity, clinical creep, derivation of the target non-inferiority margin, overcoming the blind by rating all subjects the same, etc. An ambitious question, but Mark, do you want to take a stab at that one?
Sure. No, it's a very interesting question. I hope I don't risk giving a whole lecture just to address this. But I did talk to, and the basic example was a null. There was no different drugs, and you're trying to prove that. In that sort of setting, let me bring the result null. So if you're trying to demonstrate the drug has an authority setting, things are kind of reversed. And first, the second, reviewing what non-inferiority is for those who might not be familiar with already, is you're trying to demonstrate that your drug is not much worse than an active agent. So if an active agent is known to have an effect, and basically an approved drug, and you show you're not much worse, that might be sufficient in certain settings to demonstrate your drug does have efficacy. But the challenge is the more noise you add to the system, whether that sloppiness in endpoint ascertainment or in following the protocol, more likely the two drugs will look the same. That's actually what you're trying to demonstrate. So for FDA, the sloppiness will actually show that the drugs are actually non-inferior, which is what the company's trying to show. So we do have a guidance on this. FDA does have a guidance on non-inferiority trials, and if you're interested in learning more about it, I suggest you review that guidance. Can people hear me okay? I just got a note about bandwidth. Leonard, can you hear me okay?
A little choppy, but going better now. Yeah, going better now.
Okay. Let me, I don't know what I could do. I could stop my video. I'm going to try that. I'm going to stop my video. So excuse me, I'll try to continue without the video and see if that helps. Is that any better, Leonard?
Seems to be
or
clear.
about the same? Okay, well, let me try to continue. So FDA does have a guidance on non-inferiority trials. So if you want to learn more about that, I suggest you read that guidance. But basically, FDA will look at the quality of the trial, how well it was conducted. At least that's one aspect of evaluating the evidence from a non-inferiority trial. Whether it was done to reduce the biases I talked about, or any sort of noise. Also, we'll look at any sort of evidence that the comparator arm has the effect that it's intended to have, and that's called assay sensitivity. And we might look at historical trials to see how stable the active control is in its effect. So this is somewhat of a complicated area, and involves a lot more care than what we call a superiority trial, where you're trying to demonstrate you're better than a control. So I do recommend that you start looking at this guidance if you're interested in this area.
Yeah, thanks very much, Mark. I think obviously it's a very complex subject, but presumably a lot of the reassurance comes from situations where the control drug has a large effect size, so you're not likely to make a large error with the investigational drug. But I'm sure there are a lot of other considerations that, if we had time, we could expound on. Let's see, I'm going to look for another one for Shabnam here. Shabnam, there's a question for you about what must be included in the investigator brochure. If I'm understanding correctly, what the question is. Any thoughts there?
Yes, hi, Leonard. Can you hear me okay?
Yeah, fine.
Okay, great. All right, yeah, so thank you for the question. So that is on our fda.gov if you look up investigators brochure, but briefly, the IB is expected to contain a brief description of the drug substance and the formulation, If you know the structural formula, that can also be put in there. It usually includes a summary of the pharmacological and toxicological effects of the drugs in animals, and to what extent, if they're known in humans as well. It'll have a summary of the pharmacokinetics and the biological disposition of the drug in animals, and then again, if known in humans. And then importantly, related to my talk, not that the others aren't important, of course, but a summary of information relating to the safety and the effectiveness in humans. And that's usually obtained from prior clinical studies, which are really very informative. And then they also include, of course, the description of possible risks and side effects that are to be anticipated on the basis of prior experience with the drug under investigation. Also, like I mentioned, if there's any risks and side effects that are associated with related drugs, that information would also be in the investigator brochure. And precautions or special monitoring to be done, that could be included. So those are some of the content that's expected to be in the IB.
Right, thanks very much, Shabnam. And just for everyone's reference, tomorrow afternoon, the concluding session deals primarily with material that's in the investigator brochure. So I would certainly encourage you to attend that session. Shabnam, while you're on, another question for you. The question is, if death is the endpoint, do you report death as an SAE via MedWatch form? Do you have any thoughts on that?
Yeah, we didn't talk about reporting so much, there is a form, I think it's 3500A, I think it's equivalent to the MedWatch, but for that question, I'd refer the viewers to the guidances, because they're very explicit on the reporting of the time, and I know there are questions about the 24-hour period and business hours and things like that. I would encourage you to go to the references where it's the sponsor's responsibilities. It's a draft guidance that would have that information. But I did want to address one of the other questions, because I think there were a couple of questions related to this, and that was that what gets reported by the investigator to the sponsor. So an investigator must immediately report all SAEs, whether or not the event is considered drug-related. And then again, as I said, they should also include that assessment of whether there's a reasonable possibility that the drug caused the event. But one of the exceptions to that are study endpoints that are SAEs. Those would just be reported in accordance to the protocol. But the exception again to that is that if there's evidence of a causal relationship between the drug and the SAE, even if it's a study endpoint, then in that case, the investigator has to immediately report that SAE to the sponsor. So I think we had, that previous example on my slide where, the 75-year-old had died in the trial. Death was a study endpoint, but then we later learned that the death was due to anaphylaxis. And, when you hear that term, that would raise a red flag. And there's evidence with that PT that suggests a causal relationship between the drug and the event. So in that case, the clinical investigator must immediately report that particular event to the sponsor. So I hope that clears that up.
Great, thanks, Shabnam. I know we're coming to the end of our time. I had another question for Mark, which I think raises an important issue about what you do before the study's already run. The question is, at what stage should we submit the statistical analysis plan to the FDA at the same time as the clinical trial protocol, or is it possible to only submit the statistical analysis plan at a later stage, as long as before the data analysis? Any comments on that, Mark?
Yeah, sure. No, it's a good question. Typically, for a blinded study, we don't require the SAP submitted at the same time as the protocol. The protocol should have enough information on the broad outlines of what the analysis will be, what the endpoints are, what the hypotheses are, what the statistical method should be in sort of broad terms. The specifics of implementing that, which is covered in the statistical analysis plan, is typically not submitted at the same time as the protocol, and can be submitted as long as the data has not been unblinded. If there could be assurances that the data is still blinded, we sometimes see APs rather late in the conduct of a clinical trial.
Thanks. I'm thinking that we can just squeeze in a last question. There's a question here on at what point in the study will you set your alpha, pre-study or midway? I think it pertains to the same thing, how you do things before the study evolves, and are there any consequences? So at what point in the study do you set your alpha, Mark?
Yes, that would probably certainly be something that would be done at the protocol stage, because that kind of affects what level of evidence we expect at the end of the study. So I think typically, FDA and the sponsor would like to have some agreement on that before a study commences, because it may have effects on the study size or the length of the study. So that is, I think, something that's usually decided very upfront.
Great. I think, if I'm not mistaken, we've run out of time on our panel. So thanks for two great presentations, very important topics in our process of drug approvals and therapeutic approvals. And of course, thanks to the audience for your enthusiastic participation. Sorry we couldn't get to more of the questions.